All right, everyone, welcome back. So this next session, we'll have our first presenter, Danielle from Carnegie Mellon University. Her field is neural computation, and her advisor is Brent Doran, okay. And she did a practicum at Lawrence Berkeley National Laboratory in 2015, and her presentation is on relating visual working memory computations to network architecture in the brain.
Thank you. So I'm going to begin actually by giving you a little bit of a broad overview about why we would want to relate any sensory motor computations to network architecture in the brain, and more broadly tell you why I think we really need modeling in neuroscience, why we need theoretical neuroscience, especially because that's something that this fellowship really pushed me to pursue when I wasn't pursuing it before. So I think a lot of what we're trying to do as neuroscientists are understand the computations that underlie sensory motor behaviors. And there's a whole range of sensory motor behaviors. At the top, you see one of the first projects that I worked on in my PhD, which was a human brain-machine interface. And in that case, perhaps it's a little bit more obvious that you have neural information that's driving your movements. Maybe you don't think about that all the time, but clearly when you have an array hooked into a patient, it reminds you that all of the arm movements that you make, all of the reaching and grasping, they're all neurally guided. And similarly, all your incoming sensory information has to be interpreted by your nervous system. So below is a little bit of work that I did with Chris Bouchard at Lawrence Berkeley National Lab. We were trying to understand how the auditory system tells apart different tones and what the tuning in the auditory system looks like. And so in that case, your nervous system has to disentangle the different frequencies that you have coming in and make sense of the sounds that you're hearing in the world. I don't know why this isn't working. And so I would argue that we always have to start with as I said, a sensory motor computation. Let's say that you want to understand a sound that is incoming. The first thing that you might do from a nervous system level is look at the response of a single neuron. So let's say if I was exploring this in auditory cortex, I might look at the firing properties of a single auditory cortex neuron and see that it prefers a tone of a certain frequency. But really, I would argue that we're interested in the coordinated activity of populations of neurons, not just single neurons, because that's how they really work together to be computing units. How does their firing evolve together over time? And even though we might be observing the activity of hundreds or thousands, or the whole brain has 100 billion neurons, is there some low dimensional structure that is underlying the evolution of their firing rates over time, because that'll make it much easier for us to understand what they're computing. I think that the last question, once you have looked at kind of firing responses in the brain, is what is the cellular circuit that is responsible for creating those firing responses? And this is something that people have been interested in for a very long time, as early as this is a sketch from Ramon y Cajal. And because people really want to know what the mechanistic underlying basis of these neural computations is, and a good way to get at this is from the cellular stage up. However, we're still kind of at a point in neuroscience where it's very hard to do this with actual data because the recording technologies that we have only allow us to record maybe 100 to, we're finally breaking 1,000 neurons at a time with high fidelity. That's vastly undersampling any even microcircuit in the sensory system that you would want to understand. And so I'm going to argue that the best way to do this in the modern age is to build network models. That's what we like to do as computational scientists, and that will really allow us to probe these networks and interpolate the parts of the networks that we couldn't see with recording technology. And so I am going to do a particular case of this today. I'm going to tell you about a specific sensory motor computation. It's a spatial working memory task. In this particular task, a non-human primate is asked to first fixate on the center cross. Then a cue is shown at a particular spatial location on a screen. Then there's a 500 millisecond delay period, and then the primate is asked to make a saccade, which is an eye movement, to the location where that cue previously was. And that might sound really easy, but what has to happen in order for the primate to be able to make that eye movement is that the primate has to remember or really store an encoding of where that visual cue was, even when it's left the screen. And how do you maintain a code for something that's no longer in front of your eyes? And if we look in the prefrontal cortex of the brain, we can record there during the delay period, and we see that indeed this is where that working memory cue is held, because single neurons have preferential responses for visual cues at certain spatial locations. And then if you look at the whole population of neurons, they show a bump of activity at the place where that spatial cue was during that delay period, and that's how you maintain a persistent representation of the visual cue even over the delay. So tasks like this have been done before, but there's something a little bit special about the data set that I'm working with. For one, this task is usually done in a single dimension on a 1D ring, and in this particular case, the person collected it in 2 dimensions, so the primate can look to any of the 40 spatial cues that you see on the screen. And so no one's really done a bump encoding model of working memory in full spatial 2D. And the other special thing about this data set is that they simultaneously recorded from V4, which is kind of the last area of visual processing before that information hits prefrontal cortex. And so you're really seeing multiple stages of this network, which if you're trying to gauge how a circuit works is extremely important. And so first, or then let's look at, we talked about the next step is to look at the statistics of the firing of these neurons during the task. What are the population dynamics? And when we observe the dynamics of neurons in a single hemisphere of prefrontal cortex during this task, because you can only record one hemisphere at a time, we see that neurons that prefer visual cues across the entire spatial field, that have preferred orientations on both visual hemifields. And that's notable because we see this specific weird hemispheric effect, mainly that even if 2 neurons have preferred spatial locations that are very close to each other in physical space, if they lie across the hemifield line, as those 2 purple dots do, the correlation between their firing activity is extremely low. And this is only true when they lie across the hemifield line. So there's something weird happening architecturally here, and that starts to hint at the fact that the wiring diagram in this circuit matters. Moreover, like I said, we're interested in how coordinated populations of neurons work together. So the next thing you might want to do is, try to see if there's any low-dimensional structure in this data in either V4 or PFC. And one way you can do that is with factor analysis. Factor analysis is essentially a dimensionality reduction technique that allows you to partition a covariance matrix into an independent component, which is representative of the independent Poisson-like firing behavior of every neuron in the network. Neurons are inherently going to have noise. That's different than what we care about. We care about the shared covariance, which is what are the kind of underlying low-dimensional latent structures that are driving the entire population together. That's what would be of interest to us. And if you do this in both V4 and PFC, you see that V4, and this matches up with previous experiments that people have done, does actually have very low rank structure. In fact, you can explain most of the variability in the V4 population data with actually a single latent dimension. When you look at PFC, this is simply not true. You need very many latent dimensions to explain the activity of this data. And this starts to, again, hint at some interesting circuit properties because V4 is feeding in directly into PFC. And so we start to get the sense that the wiring in PFC must be somehow filtering that low dimensional variability from V4 and creating something much higher dimensional. As a quick aside, because we wanted to talk a little bit about supercomputing here, neuroscience data is getting bigger and bigger. Do you want to perform something like factor analysis on a really large neural population? The Allen Brain Institute has this brain atlas now that has tens of thousands of neurons at once being imaged with calcium imaging. Or maybe you actually just want to perform factor analysis on the output of a very large model that you make, which isn't even real data, because that's a perfectly valid thing to do. And so we noticed, me and a couple other machine learning students at CMU, that no one had really done this for factor analysis, made a large, a publicly available, scalable method. And so we created scalable factor analysis in Spark, and that's something I wanted to bring up for this fellowship because I feel like it's something that I never would have done previous to this. And so I don't want to go through all the details here exactly, but essentially what factor analysis can be solved using an expectation maximization algorithm. And there are parts of that algorithm that we can optimize using Spark. Specifically on the largest matrix computations, we could use Apache Mahout for efficient row out or product formulations of those problems. There are times when you can leverage matrix sparsity in these computations. And specifically, I've put kind of little paper airplanes next to all the places where it was worthwhile to send this stuff out to Spark servers. And so our method performed nearly as well as probably the closest publicly available dimensionality reduction technique, which is a Spark application of probabilistic PCA. It's a similar type of approach, but there are different ways that you optimize those. In terms of the theoretical time bound, our algorithm should actually be better. We probably didn't spend nearly as much time optimizing it as the SPCA people did, but we got real close considering this was mostly a thing that we did for ourselves. And we were running this on Amazon Web Services with Allen Brain Atlas data. But back to the kind of modeling constraints. So we've learned, again, some weird constraints from the population data that we were looking at before. Mainly, you go from low dimensional structure in V4 to high dimensional structure in PFC, and we see this strange hemispheric effect across PFC, which tells us that there must be much stronger recurrent connections within hemisphere in PFC than across hemisphere in PFC. And so what my interests are kind of now in the late stage in my PhD are in developing really mechanistic models of circuits, starting from cellular properties, mainly that all neurons fire action potentials. And one of the easiest ways that we can model this is to develop a spiking neural network where every neuron is essentially modeled as what we call a leaky integrate and fire unit. It's essentially a 2D differential equation RC circuit with a leak in it, which is pretty tractable to do actual computations about. And then basically we put neurons into broad classes like excitatory or inhibitory. Every neuron in the nervous system is one of those two things that either increases another neuron's current input or decreases it. And the goal is to connect these simple spiking units with generalizable network wiring rules that are only based on properties like cell type or the tuning specification of a cell, or as I'm about to show you, this concept of assemblies. And the whole idea about doing this is to develop intuitions about the population dynamics that we can start to explain with these kind of mean field approaches that you might steal from statistical physics. We don't want to think of every neuron like a snowflake. We want to have some generalizable model across the population where you can very easily explain things based on some broad wiring rules. And so this is what I'm starting to get into now. Essentially, I have created this network that has these assembly-like structures where you see the every excitatory neuron is represented here with the triangles, inhibitory neurons are represented with circles, and they have clusters now, and they're more likely to connect to other neurons within their own cluster than they are to connect across clusters. And if you know anything about broader spiking neural network theory, we have this thing that we can do called balance networks, where essentially we balance the input between excitatory and inhibitory neurons. This is really important if you want to create networks with internally generated variability. And considering we wanted to study how variability was filtered through this circuit diagram, how we got this change in variability from V4 to PFC, we do want the recurrent architecture in our network to actually generate variability as opposed to having to add noise to the network. But now that we've added these assemblies, we get something that we wouldn't get with a classic balanced network. A very traditional balanced network would have completely random connectivity, or like an Erdos-Renyi graph structure. And by putting in these assembly structures, we start to get essentially up, down, on, off states of spiking of neural behavior, where you get competition between these two clusters. And that's exactly what you want for the types of behaviors that we were seeing in PFC, where I said we had this weird idea going on across hemispheres. And so this figure that you see here, because I'm only starting to get deep into this, is actually from a previous student in the lab who did exclusively excitatory clustered connectivity. You actually get much weirder behaviors when you start clustering excitation and inhibition, but much more exciting ones, I would argue, ones that are perhaps capable of also maintaining a persistent memory representation, which is something that his old network did not do. So that's kind of what I'm working out now in my late stages of my PhD. I don't quite have those results yet, but I wanted to show you essentially the model I've been building and where I've been going. And so I hope with all of that together, I've convinced you that we're starting to get at these questions of how does network architecture produce coordinated working memory computations, and that in general, by using these types of theoretical neuroscience models, we can answer questions about how architectures are producing a vast range of sensory motor computations. And so with that, I'd like to thank my primary advisor, Brent Waran, Sanjeev Khanna, who collected the majority of the primate data that you see here today in the Department of Bioengineering at Pitt, his advisor, Matt Smith. On the bottom is my co-advisor, Valerie Ventura. She's in the Department of Statistics. She helped a lot with my first PhD project, which was in neuroprosthetics. You saw Jen Schuerman controlling the BMI earlier. That was data that was collected by Rob Gaunt in the Rehab Neural Engineering Lab. I did my practicum on auditory models with Chris Bouchard, as I mentioned, at Berkeley National Lab. And of course, I would like to thank the CSGF and Krell Institute for, like I said, pretty dramatically changing the path of my PhD. I went from very applied biomedical engineering work to this quite theoretical neuroscience modeling work. And with that, I'll take questions.